Good morning, good early afternoon, everyone. My name is Susan Howell. I am the genetic counselor and clinic coordinator for the Extraordinary Kids Clinic and Children's Hospital, Colorado. And with me today is Kayla Molison, who's the genetic counseling assistant at our clinic and also a research service professional with our team. So today we're going to be presenting on understanding X and Y variations from a genetic diagnosis perspective, the terminology that many of you have probably heard throughout the course of the weekend, and then next steps planning. So to start, we'll talk about some of the important terminology. And I apologize because probably this talk should have been in the earlier part of the weekend. But when we back up and we're talking about chromosomes, we're talking about the actual structures in our cells that carry the DNA. Typically, people have 46 total, one copy in each pair from each parent. So one from the dad and one from the mom. Aneuploidy is a term that refers to having additional or missing chromosomes in a cell, so an abnormal number of chromosomes. And when we talk about sex chromosome aneuploidy, we're talking about having an additional or missing X or Y chromosome in the cells. Commonly, there are kind of the most common two buckets, the sex chromosome trisomies, meaning that there are 3 copies of the sex chromosomes. These can refer to Klinefelter syndrome or XXY, Jacob's syndrome or XYY, and then trisomy X. But we can also see commonly sex chromosome tetrasomies, tetra being 4 sex chromosomes, where we have XXYY, 3XY, or 4X's are examples. So when we're talking in a karyotype or a chromosome report, we commonly talk about 47 XXY. 47 is referring to the total number of chromosomes in the cell. Instead of the typical 46, there's 47. The XXY is notating which sex chromosomes are present in the cell, and XXY is notating that there are 2 X's and 1 Y chromosome. And a lot of these conditions have many names, but for all intents and purposes, they're referring to the similar conditions. So a scientific term that's commonly used in the literature is sex chromosome aneuploidy because it's very descriptive from a biologic perspective, or SCA. But we also see terms of abnormality, anomaly, disorder, and variation. Is the X and Y chromosome description. So we can see X and Y chromosome aneuploidy, and then families often prefer X and Y chromosome variations, which we'll try to embed throughout today's talk as well. So the bottom line being is there's a lot of different X and Y variations. This picture represents the different variations that we can see, also reflected in the size of the circle by being how common the trisomies are in comparison to some of the other aneuploidies. And the incidence that we can see is also quite variable. The most common conditions in males are XXY occurring in 1 in 650, and XYY occurring in approximately 1 in 1,000 males. And then in females, trisomy X being 1 in 1,000. And the more chromosomes that are present in a cell tends to have a lower prevalence for those conditions. They tend to be more rare. And combined, when we're talking about all of the X and Y variations as a group, they're actually quite common, being around 1 in 400 live births. So how do we identify X and Y variations? Historically, it was from congenital abnormalities or birth defects would have genetic testing as part of the workup. We also historically would do genetic testing when a child presented with global developmental delay or hypotonia with significant low muscle tone, or a diagnosis of autism as an indication to order genetic testing. And often physicians are trained to order genetic testing in a child who presents with more physical symptoms or physical differences in their appearance. However, historically, these conditions have been underdiagnosed. And many of the features that we can see in these conditions, such as ADHD and anxiety, like was presented before by Sierra and Caitlin, they don't actually serve as indications to order genetic testing. So even in the children that are undiagnosed, it doesn't mean that they don't present with symptoms. It just means they don't come to medical attention to do genetic testing. There's a relatively new technology that you may have heard mentioned throughout the weekend called prenatal cell-free DNA screening, also referred to as non-invasive prenatal screening. And this is a technology that's now identifying X and Y variations during pregnancy today. So this technology is analyzing fragments that are from the fetal DNA, actually with an origin of the placenta, and these fetal fragments of DNA are naturally occurring, found in the mother's blood. And so it's a maternal blood test done during pregnancy that measures these fragments of fetal DNA. And so this testing is a screening test, right? It's not a diagnostic. Other terms that are commonly used is NIPS, non-invasive prenatal screening, or NIPT, non-invasive prenatal testing. It's all the same stuff. It became commercially available in 2013. However, the American College of Obstetrics and Gynecology started recommending for all pregnant people, regardless of their age or prior risk, as of 2020, should be offered this screening. And we're seeing more and more families being identified through this screening. It's performed as early as 10 weeks gestation, and that 10-week is really the timing in which there's a high enough concentration of fetal fragments in mother's blood to be able to actually conduct the test. And results of this screening can be either a positive screen, a negative screen, or an inconclusive screen. It's not diagnostic as a screening test. It only identifies a risk for an aneuploidy. And we do see that there can be some risk of discordance in which the result from the screening is actually different from the actual result from diagnostic testing, which I'll explain about a little bit in a minute. So as this technology has come online, it's really been widely adopted because it doesn't pose any risk of miscarriage in the pregnancy. We are seeing that the targeted testing in this screening is actually for not only X and Y chromosome variations, but also trisomy 13, so chromosome 13, chromosome 18, and chromosome 21. However, follow-up testing is always recommended because it is not diagnostic as a screening. And when we talk about the ability for this screening to actually identify a positive result with an actual diagnosis, we're referring to positive predictive value. So what's the likelihood or the probability that a screening test that's positive is actually consistent with the diagnosis? And we can see that can be pretty poor in the X and Y variations in this technology. So a couple of years ago, the American College of Medical Genetics published a meta-analysis of 28 studies looking at all of the X and Y outcomes and the positive predictive value, which is highlighted is a true positive in the child or in the pregnancy. And overall, this is about a 43%. And breaking it down by individual conditions, Turner syndrome, also referred to as 45X, has the poorest positive predictive value. And then we see a little bit better with trisomy X and a little bit better, but even at best, we see about 74% positive predictive value in XXY and XYY. So there's definitely a big gap when you get this type of result. We've also seen through the Extraordinary Babies study several cases of discordance. This is a table that was taken from a publication in which we had 12 cases where the prenatal cell-free DNA screening had identified a risk for having a trisomy such as XXY or XYY, and the family was counseled on that risk. However, diagnostic testing, either done prenatally or postnatally, confirmed a diagnosis of a tetrasomy. And in this case, it's 48 XXYY. And many of these children are actually being followed through our extraordinary baby study as well. So when we have a diagnostic, when we have a screening result that is positive or at risk for having an aneuploidy, diagnostic testing is recommended. This can be done either prenatally during the pregnancy or after birth. And what we see is about half and half, that families will elect to either do prenatal testing to confirm the diagnosis while pregnant, and half the families will defer the testing until after the baby's born. And most likely that's because of the fact that they're the prenatal diagnostic testing puts the pregnancy at risk for a possible miscarriage. It's a relatively small risk, but nonetheless a risk. So what we commonly see in prenatal diagnostic testing is either a CVS or a chorionic villus sampling that's done at 10 to 12 weeks, but because the screening result may not be back until after 12 weeks, most commonly we see prenatal testing with an amniocentesis. And this is a diagnostic test that can be done at 15 weeks gestation or later. And then postnatal testing after the baby is born can be done on a blood sample. Commonly, we recommend or encourage families to try to do cord blood if they can. They don't have to poke a newborn baby. You can also do postnatal testing by cheek swab or a saliva sample, although getting saliva from a newborn is quite tricky. So, and then Kayla's going to go ahead and start talking now in regard to the genetic testing methodologies that are used to confirm a diagnosis.
All right. Can you guys hear me? Perfect. So there are a few different genetic testing methods that we can use when it comes to actually confirming a diagnosis of an X and Y chromosome variation. So one of the most common types is something called a karyotype. And this is a type of genetic test that specifically looks at the number of chromosomes or the chromosome count, and also the structure of chromosomes. And so when we talk about a karyotype, in terms of how it works, we take a lab basically takes that cell sample that was received, like Susan mentioned, from something like a blood draw or a cheek swab, and they basically use a special dye that makes the chromosomes visible, and then they image that. And when you do that, you get a picture that looks like the circle up above, where we can kind of see all the chromosomes and they're all stained. But this is quite messy and difficult to interpret. So what then happens is the chromosome image is basically rearranged to generate the images below. And this is what's actually called a karyotype or a karyogram. And so in a karyotype, what essentially happens is all of the chromosomes are rearranged into their respective pairs, as you can see, and then they're rearranged to be in roughly a size order from biggest to smallest. And then finally, the sex chromosomes are put at the very end, and they're labeled with whether they are an X chromosome or a Y chromosome. And that's essentially makes it a lot easier for interpretation of the actual chromosomes themselves. And so if you look on the left, we have 46XY male. And so this is a result where we see that this individual has 2 copies of each chromosome. So 1 copy from each parent, and then 1 X chromosome and 1 Y chromosome. And then right next to that, we have an example of a karyotype from a 46XX female. And as you can see, we have 2 copies of each chromosome, 1 through 22, and then 2 copies of the X and 0 copies of the Y. So when it comes to actually doing karyotypes for people with X and Y variations, we see a little bit of a different result. So as you can see, this individual has 2 copies of chromosomes 1 through 22. And then when we look at the sex chromosomes, they have 2 copies of X and 1 copy of Y. And so this particular karyotype result would be diagnostic of 47XXY or Klinefelter syndrome. When it comes to the other conditions, for instance, this particular individual has 2 copies of chromosomes 1 through 22, and then 1, 2, 3 copies of the X chromosome, and then 0 copies of Y. So this is a result that would be diagnostic of 47XXX or Trisomy X. And then finally, for the last trisomy, this individual has 2 copies of chromosomes 1 through 22, and then one X chromosome and 2 Y chromosomes. So they have XYY, which is diagnostic of 47XYY. But this isn't the only method that we can use to do genetic testing, although it often is the most common. Another method that we could use is something called a microarray. And so a microarray looks a bit differently and also works differently from a karyotype. And so with a microarray, we essentially have many thousands of probes that attach to very specific places on each chromosome. And they can basically bind and attach. And then what can happen is the lab can analyze how much chromosomal material is attached to each probe. What they also do during that analysis is they can come back with more attached chromosomal material or less attached chromosomal material than we would expect. And what that can help us do is identify breakpoints, deletions, and duplications, and including aneuploidy, so basically extra or missing chromosomes. And so I have a picture down here below that's a little bit complicated, so I'll kind of break it down. But if you see this big red line going across the numbers 1 through 22, that is indicating that there is a normal representation of the chromosomal material that we'd expect for those chromosomes. So these all have 2 copies. However, when we get to the X chromosome, you can see that that red line is a little bit higher. And that's indicating that there's an overrepresentation of material from the X chromosome that attached to the probe. So something like this would be diagnostic of trisomy X. And that's how a microarray works. Now, this is also a good point to get into something called mosaicism. And mosaicism is a little bit hard to understand at first, so I'll kind of break it down. But this is when there is the presence of multiple genetically different cell lines within one individual. And so we have an example of how this might work in this figure below. And so if someone is non-mosaic, it basically means that all of the cells in their body are genetically the same. So for instance, if someone has non-mosaic trisomy X, all of their cells have 3 X chromosomes. But we can have mosaicism in which the results are different. So in the first picture with the blue and the orange, you can see that there are a mixture of cells with 3 X chromosomes. And there's also a mixture of cells with 2 X chromosomes, so 46XX. So when we have genetically different cells like that, that's mosaicism. We can also have a result with 2 different aneuploid cell lines. So as you can see, we have in the picture on the far right, we have trisomy X cells with 3 X chromosomes, but then we also have a Turner syndrome cell line with just 1 X chromosome. And so this is proportions that we have seen clinically in some of our patients. And so it's important to note that mosaicism can happen for a lot of patients. One common one common belief is that if you are mosaic, you often will have milder symptoms. I want to emphasize that this is not always the case. It's a little bit more complicated, and the prognosis depends on what exact cell lines are present. And so if you do have mosaicism, it's important to talk with someone like a genetic counselor or a geneticist just to better understand what follow-up care should look like. It's also important to note that it is possible for different parts of the body to have different ratios of cell lines or different levels of mosaicism. So sometimes what we see is that if we test 2 different samples from different parts of your body, say blood and then a cheek swab, sometimes the levels of mosaicism will actually come back different. So that's something that can also happen. It is important if mosaicism is identified on something like a karyotype to have follow-up FISH testing, because this is a bit more sensitive for detecting low levels of mosaicism because it looks at more cells. And so that's something I want to get into now. So FISH testing, which stands for fluorescent in situ hybridization, is another method that we can use to confirm the diagnosis. And essentially what this does is it uses probes that are capable of fluorescing or basically glowing. And these probes attach to specific places on specific chromosomes. And so because of that, we can actually identify what chromosomes are present and how many of them are present based on the number of glowing dots. So for instance, if we have a probe that binds to the X chromosome, if we see 1 glowing dot for that probe, that means there was one X chromosome present within that cell. If we see two glowing dots, that means there are two X chromosomes within that cell. And so in FISH testing, we often evaluate 200 or more cells, which means, which is a lot more than you would do in something like a karyotype, which means it's a lot more sensitive for identifying mosaicism, and also can detect really low levels of mosaicism, which is why it's advantageous. About what exactly causes X and Y variations. And so to put it simply, X and Y variations are caused by something called chromosomal non-disjunction. And this is when chromosomes fail to separate properly during cell division. And so when we talk about cell division, there are basically two methods that we're talking about. One is meiosis, which is used for the generation of sperm cells or egg cells. And one is mitosis, which is used for just general growth and repair of your body. And during these processes, DNA basically gets duplicated and then it gets split evenly between the daughter cells. So we start with a parent cell that's going to divide, and then we get the daughter cells that split that material equally. But if we have an uneven split between daughter cells, that is what non-disjunction is, and that can result in X and Y variations. I want to emphasize that this does not happen because of something that a parent did or did not do before or during pregnancy. Non-disjunction is random, and so at no point should anyone ever feel like it was their fault that this happened. So I know that it's kind of easy to talk a little bit about meiosis and mitosis, but it's harder to kind of have a good understanding of what all those words mean. So I wanted to kind of show you how this works with one of my favorite things, which is PowerPoint animations. I'm absolutely obsessed. So this is what typical meiosis looks like. And so we essentially start with a sperm or egg precursor cell. So this is a cell that has 23 pairs of chromosomes, one from each parent, or 46 total. And they have a pair of sex chromosomes, which are represented below in purple and green. Okay, so the first thing that happens in meiosis is something called replication. And so each chromosome is going to get copied. So if you watch the green and purple, you can see another one basically appears right next to it. And then what happens is we go through something called meiosis 1. And so meiosis 1 is when the cell divides into two and splits the chromosomes equally between the daughter cells. Then what happens is we go through something called meiosis 2, in which the cells divide again and split the chromosomes equally. Now, at this point, we basically end up with a cell that has one copy of each chromosome. And this might seem a little counterintuitive because we are all supposed to have 46 total, two copies, right? But remember, all of these cells are going to get combined with a sperm or egg cell. So they're going to combine to get two pairs. But at this point, all of these sperm or egg cells just have one copy. And so these are all ready. These all have one sex chromosome, and these are ready to be combined with an egg or a sperm cell to make a zygote. Now let's talk about non-disjunction and what that looks like using those same animations. So again, we start with a sperm or egg precursor cell with 23 pairs of chromosomes, including a sex chromosome pair. We go through replication, same as always, the chromosomes get copied. But this time, we might have non-disjunction in meiosis 1. And in this case, the chromosomes don't separate equally. And then when it goes into meiosis 2, they split the chromosomes equally, but these two end up with an extra sex chromosome. And so if this is combined with an egg or a sperm, there will be 3 sex chromosomes, which is a trisomy. So this is one way that we can actually see trisomies happen, and this is how non-disjunction generates them. And so it's also possible for non-disjunction to happen in meiosis 2 in that second step. And it's very similar, but I'll walk through it again. And so we start with that same precursor cell. We have replication. The chromosomes get copied. And then this time, meiosis 1 works correctly. And so it splits in two, divides chromosomes equally. But if we have non-disjunction in meiosis 2, as you can see with those bottom two, the chromosomes weren't split equally. And so once again, this cell has 2 X chromosomes. And so if combined with an egg or a sperm, it'll get another one. It'll have 3 total, which is diagnostic of a trisomy. So that's just a little bit of an actual more clear example of how this works, because I know it's hard to visualize. Okay, And so just to finish up on a last few things for my part, but I also wanted to address how exactly mosaicism happens. And so mosaicism is a little bit differently. It's still caused by non-disjunction, but instead of happening during meiosis, it happens during mitosis. And mitosis is after conception. And so with conception, you get a zygote. And so if it happens after the zygote is made, it's called post-zygotic non-disjunction. And so I think this figure here does a good example of kind of showing how this might work. But as you can see, the sperm and egg both have one copy of an X chromosome, or sorry, a sex chromosome, and they come together to make a zygote with a normal number of sex chromosomes. But somewhere a little bit down the line, we have a non-disjunction event, as you can see with the green. There's now a cell line that is basically missing a sex chromosome. And this cell will continue to get copied during mitosis and will generate more cells that are aneuploid. However, this didn't happen at the very beginning, right? We start with that first fertilized egg, that first zygote. It does have a typical number of sex chromosomes, and that is continuing to grow throughout the organism, right? And so we actually have two different cell lines that are basically continuing to grow. And because of that, at the end, when we kind of look at the whole organism or the whole body, we actually might end up with a scenario where some of the cells have a X and Y chromosome variation or are aneuploid, and others are not. So I think this last image here does a good job of showing how that might work. All right, and just wrapping up, based on all the genetic testing things we've discussed today, there are some common counseling points that we want to emphasize. So as Susan has mentioned, it is possible to have a different result. NIPT can indicate that you might have an X and Y variation, but we can see that that might not be the case, or it might actually be a different X and Y variation. And so a cfDNA test or an NIPT always needs to be confirmed. Also, it's really important to keep a copy of the lab report, right? People have different conditions, and sometimes it's really a little bit more complex. You might be mosaic, you might have a karyotype, and then FISH testing that identifies different levels of mosaicism. There can be a lot going on. And so it's important to keep a copy of the lab report so that you or your child has it for future medical purposes. Additionally, I just want to emphasize that couples who do have a child with an SCA typically have less than a 1% chance of recurrence. And so and these conditions are very rarely inherited or passed on, but with rare parental chromosomal conditions, we know that it can happen. And so it's always important just to talk to a genetic counselor just to better understand what future risk might look like, even though these are rarely inherited or passed on. Okay, so I'll pass it back to Susan now.
Give Kayla a round of applause for the animations. I think she did a fantastic job. Okay. Now I can see. Okay, so we're going to talk a little bit more about, when we have these conditions, how does that relate to, from a chromosome perspective, relate to the child and some of the symptoms that we can see. And so we recognize that the size of the chromosomes that carry our DNA often reflect the number. You can see in this depiction that the X chromosome is relatively large. It carries a lot of DNA, especially when you compare it to the smaller chromosomes, and the Y chromosome being fairly small. And when we're understanding chromosomes and what it means to have an extra X or an extra Y, not only are we, studying the structure of the chromosome from a karyotype or a microarray, but then recognizing that the DNA coming in base pairs, the X chromosome having 153 million base pairs. And you're not supposed to be able to read this slide. That's actually by design, but this is a list of conditions that can be affiliated with mutations on the X chromosome, right? And that is huge. There's a lot of impact that can happen from differences on the X chromosome. And so it's complicated, right? This having an extra X is really complicated because of this. 50 million base pairs and several conditions that can even be associated with a much smaller chromosome. So I'm going to talk about some, biological concepts that have been discussed throughout the course of the weekend, just for clarity, as well as kind of helping us from a hypothesis generating perspective and understanding what's causing what. So X inactivation is a process in which Mother Nature tries to balance out in the cells, males and females. So if a female typically has 2 X chromosomes and a male has 1 X chromosome, Mother Nature. So that second X chromosome gets inactivated so that genetically it's somewhat balanced. It's a random process. Which X chromosome is inactivated, and it happens early in embryonic development. However, not 100% of that inactivated X chromosome is actually turned off. And so an example of that is there are these regions on the tips of the chromosome referred to as pseudo-autosomal regions, meaning that they almost look like the numbered chromosomes, not the sex chromosomes, where there's copies on each. And these areas escape X inactivation. They're not inactivated on the X chromosome. And it's thought to be that these overexpression of areas that escape X inactivation may contribute to some of the symptoms and phenotypes X chromosome or an extra Y. So when we're talking about X chromosome genes that escape an X inactivation, you can see from this picture, which again, you're not really supposed to be able to read, but it's highlighting all the areas of the X chromosome, the 12 to 15% of the X chromosome that can escape X inactivation. And that means that these genes continue to be expressed from the inactive X. And so when you have extra X chromosomes, then you have extra expression of these genes, right? Because the more X chromosomes, the more gene expression. And then this also applies to Y chromosomes because of the fact that they're not inactivated. So having an overexpression of those genes when you have an extra Y. Another prior hypothesis has been that there is a gene on the X chromosome that makes a protein that's called the androgen receptor protein. It's the androgen receptor gene. And within this gene is a polyglutamate CAG repeat sequence. And it's thought to be that this CAG repeat length could be related to the effectiveness of the receptor. So this has also been something that's been commonly discussed. But more recently, what we're realizing is this is much, much, much more complex, that we're seeing that it's almost not just about the X and the Y chromosome or the overexpression of the genes just from that extra chromosome. What we're seeing is that the other chromosomes, so the numbered chromosomes called our autosomes, also get impacted when there's an extra X or an extra Y. In fact, we can see that there's a 21% of the expressed autosomal genes can be impacted in the presence of an extra X, even though it's inactivated, or an extra Y chromosome copy. And so this depiction with the red and the black dots reflects that even when we have an extra Y chromosome in this individual, that all of those extra red dots above 0 are representing overexpression of genes throughout the entire genome. So this is what we refer to as an epigenetic impact, where it's affecting all of the genes in the genome. And then red dots that are underneath the 0 representing underexpression or less than expression. So what I put up the picture of the dominoes, thanks to ChatGPT, trying to demonstrate that it really is almost like this domino effect, right? We have the presence of the X chromosome, but then that presence kind of goes out and branches into other areas of the genome, causing certain genes to be upregulated or downregulated. And it really is causing this bigger effect throughout the genome, not just based on the genes that are present on the X or the Y. So it's an exciting time in science, and we still have a lot to learn, but it is definitely not simple. So, appreciating that there's probably a lot of biological factors like epigenetics, metabolomics, which is the process of the cells to turn on or off genes, metabolomics, in which the byproducts of metabolism or proteins are being impacted on the genome, transcriptomics, as far as, certain genes turn on or turn off during development and how that can impact the genome and the expression of the impact, different proteins, microbiome. And then we also appreciate that there's a lot of environmental factors that can impact the variability that we can see in these conditions, from the prenatal environment, nutrition, education, physical activity, and healthcare. And likely what I anticipate, it's going to be more of a mathematical equation in which there's really a combination of multiple factors that really explain the variability that we can see in these conditions. But it's important, as Kayla mentioned before, that we are more than just our genes, and the biological and environmental factors do have an influence as well. So I'm going to pivot here because I think, while we can talk about a lot of the science and understanding what we're learning and maybe an explanation for why we see symptoms that we're a family and we have a child and we're trying to help the health of the individual and the development and making sure that we have the best outcomes that we can living with the condition. And so one thing that's important to recognize is that when we have a new diagnosis, we need to plan for next steps, right? A diagnosis often provides a kind of a roadmap in which we, can try to monitor for things that can be associated with an X and Y variation, but it doesn't define who the child is. The child is the same child they were before the diagnostic testing was performed. And so recognizing that as an individual, children and individuals have strengths and weaknesses. And we often will seek out evaluation for our patients after a diagnosis based on a lot of variabilities. It's not a one-size-fits-all approach. It can be based on the individual's age, the presenting concerns, and their history. So we're often evaluating for things as far as physical features and medical features, developmental features depending on the age of the child and their history and presenting concerns, and then psychological features like learning and behavior. The picture is to demonstrate from a publication that we did more than a decade ago, but really describing in all of the colored columns that the various disciplines that we have as part of our team. But commonly when we're supporting kids who have these conditions, as far as development and wanting to evaluate development, this can be done with a developmental pediatrician or a child psychologist or early intervention team. When we want to evaluate speech and language with a speech pathologist, evaluating motor skills is commonly done with an occupational therapist or a physical therapist. Learning and behavior is done with evaluations from a psychologist or a neuropsychologist, and then pediatric and evaluating pubertal progression. And there's a lot of other resources too, but this is really demonstrating the multidisciplinary need of evaluation for patients with X and Y variations. I think it's really important to also highlight with a family who has a new diagnosis that there are a lot of strengths that we can also see in our patients and children with X and Y variations. So Dr. Talia Thompson, who's not with us this weekend, had previously published a paper about the positive character strengths that we can see in our children reported by their parents. So commonly we hear parents talking about kindness and eagerness to please, perseverance, love of learning, creativity, like Kayla had alluded to, and Dr. Middleton previously, and the teamwork. And we have a couple quotes from parents as far as describing their child. He's polite, gentle, and shares things with others. She's very creative and thinks outside the box. He is like a sponge and soaks up information he hears on subjects that he likes. And so there's a lot of positive character strengths that we hear reported from parents when they're describing their child. It's not just about risks of problems. So in summary, genetics are complicated. We still have a lot to learn. These are highly variable conditions. And so it really does need to be personalized on an individual basis. We also have a lot to learn from screening because we recognize that the underdiagnosis historically reflects the fact that there is a lot of variability, and we could learn from individuals who may otherwise not have been diagnosed prior to the cell-free DNA screening. There's a lot of family background genes and environment that impact the outcomes of our patients and children. And we want to take a proactive approach when we have a child identified, especially early or in young, so that we don't take a wait and see, but we're much more proactive in getting evaluations and addressing any kind of delays or concerns early in order to have the greatest impact. Recognizing that the diagnosis does not define the individual or the child, but more gives us a roadmap on how to care for the child and support their needs. And that we see many, many strengths in individuals with X and Y variations that we firsthand witness in the patients that we follow in our clinic. And then we'll take questions. I wanted to share this picture, though. Dr. Tartaglia received a donation of the portrait from his family of Dr. Harry Klinefelter. So it's presented in the photo, and it's hanging in Dr. Tartaglia's office, and it's a big honor. And she often will look at it and say, "What would Harry do?" So, and with that, we'll take questions.